So I'm just going to today talk about DNA and the function of DNA. So the main function of DNA is to code for proteins. DNA consists of a series of letters that are called organic bases. And if I draw that really, really simply, you can see these little rungs on the ladder. These are the bases. So a very, very simple diagram of DNA looks like that, with a series of bases that carry a particular code. So a particular sequence of letters or bases codes for a particular protein. And what I wanted to show today was how the DNA actually codes for the protein. So how does the DNA code get translated into a sequence of amino acids which then becomes a protein. So you probably know that DNA is a double helix, so it's made up of two strands, and one of the reasons that it's made up of two strands is so that to provide strength, but also to provide protection to the coding regions on the DNA. But in order to get the code, you need to expose those bases. So this double strand has to separate. Yeah? And I'm just going to draw that on the board here. So at the moment it's closed. Yeah? The code is protected in order to open up the code so that it can now code for a protein. The DNA molecule has to unzip, and this is what it does. Yeah? And of course it's an enzyme that is responsible for unzipping it. And if I draw it like this, see all the bases now in this region are exposed. This region of exposed bases is going to code for one protein. And the region of bases that codes for a protein is called a gene. Okay, so that one gene will code for one protein. In order to make a protein, amino acids have to be assembled at a special structure called a ribosome. But the ribosome is found in the cytoplasm of the cell, outside the nucleus, which is where the DNA is found. The DNA is an absolutely massive molecule, so it can't actually leave the nucleus. It has to stay there. So in order to carry the code from the nucleus to the ribosome, which is down here in another part of the cell in the cytoplasm, outside of the nucleus, it has to be copied into a smaller molecule. So let me just wipe off that label, and I'll show you how this code gets copied so that into a smaller molecule, so that the smaller molecule can carry that code to the ribosome, which is out of the nucleus in the cytoplasm somewhere down here. So what happens then is each base is attaches to another base on another nucleotide. But I'm drawing it in a different colour because actually this is not a nucleotide from DNA, this is a nucleotide from another molecule called RNA. And they all pair up, and we say that they are complementary base pairing. So, for instance, if this was cytosine, or C, this base here, this complementary base, would be guanine, or G. Yeah? If this was guanine, G, on the DNA, then the complementary base would be C on the RNA, and so on. So now you can see that we've got a brand new RNA molecule that's been formed that is complementary to the code on the coding strand of the DNA. And you can see that it's much smaller, it's only single-stranded, and it's now ready to fit through the opening in the nuclear pore and go into the cytoplasm to the ribosome. So this is our messenger. So we call it M for messenger RNA. It's carrying the code, it's acting as a messenger. Off it goes to the ribosome, into the cytoplasm, and it attaches to the ribosome, and every 3 bases codes for 1 amino acid. And of course, amino acids are the building blocks of proteins. So let's say that this was C, that was U, and that was G. That would code for a particular amino acid. Yeah? So that amino acid would be brought to the ribosome and dropped off, depending on what that triplet code was. And then another one would be dropped off, depending on what that triplet of bases consisted of, and so on, and so on. And of course, a bond is made between each amino acid, a peptide bond, and eventually the mRNA is completely translated into a sequence of amino acids. So we have a particular sequence of amino acids which is dependent on the sequence of bases in the mRNA, which has been carried from the sequence of bases in the gene in the DNA. So that's how we've got our code from the DNA to our protein. And of course, this sequence of amino acids folds up and folds again, and you get your functional protein. Just to add on, just to finish off this little tiny lesson, there are ways to actually switch on this gene being expressed, i.e. being made into a protein, or switch it off. And I'm going to use this piece of tissue to demonstrate DNA. So this is a long strip of DNA, let's imagine. Yeah? And what happens usually in the cell is that the DNA actually wraps around to form chromosome. Yeah? So a chromosome is just a coiled up piece of DNA. But of course, once it's all coiled up like that, this can't happen. Remember, the DNA has to separate out to expose the bases so that mRNA can copy the code. Yeah? And it can't happen, it can't separate if it's all coiled up. And so what has to happen is a section of the DNA has to unwind. These bits might stay coiled up, but this bit will have to unwind so that this bit can now separate so that the exposed bases can be copied, then carried to the ribosome, and so that the sequence of amino acids can be assembled. And in order to allow this unwinding to occur, we have certain chemicals that can add on to the DNA to stimulate it to unwind, and even certain chemicals that add on to the DNA to stop it from unwinding, so it actually stays coiled up and is more likely to stay wound up and unlikely to uncoil. And those chemicals can come from your environment, your internal environment, or even from your external environment. So you can have environmental triggers that stimulate gene expression or inhibit gene expression. But that's another lesson.